Hey guys, so this video is on restriction enzymes. It's the first video when we're looking at biotechnology. So our intention for this video is to know what a restriction enzyme is. And we're going to understand how they're used to cut DNA. And eventually, as part of this topic, you're going to be able to actually complete a process called enzyme restriction, where you use restriction enzymes on a section of DNA. So restriction enzymes are found in bacteria. And when bacteria is faced with an invading pathogen, so for example, a virus, they release these enzymes called restriction enzymes. And sometimes you might see them referred to as restriction endonucleases, but they're basically enzymes with a very specific role. So here you can see a bacteria, in this instance it's being invaded by a virus, and the virus is injecting its own DNA into the bacterial cell. Now what restriction enzymes do is they're enzymes that will cut DNA at specific sites. Now those sites are called recognition sites. And it's actually a defense mechanism to stop invading DNA from taking over the cell. So you can see here, we've got scissors, which sort of indicate that these are these restriction enzymes or endonucleases that cut DNA at specific spots. So it's basically to stop DNA from entering, that bacteria will actually cut them up into smaller fragments so they don't work effectively. Now, each restriction enzyme has its own unique recognition site. Now, this is a site that it will look for on the DNA, and that's where it's going to make its cut. So you can see here, for example, this restriction enzyme called HIND3 is looking for a sequence of DNA exactly like this, and it's going to cut that DNA where it says AAGCTT, and you can see it makes the cut between the first two A's on the top strand of DNA, and on the bottom strand it leaves via the two A's. So you can see here, these are just a couple of different restriction enzymes with their own unique recognition sites. Now, recognition sites or restriction enzymes either leave sticky or blunt ends. Now, sticky ends overhang. So you can see here for our first restriction enzyme, this is a popular or a common restriction enzyme called EcoR1. You can see that when we've completed the cut between the G and the A and the G and the A on the bottom strand, you can see it leaves these overhanging ends of DNA, and we call those sticky ends. If you compare that to the second restriction enzyme, the one in this orangey yellow color, you can see that it actually cuts straight down in a line, and we call these blunt ends. And you can see here again that what it results in is two DNA fragments, both with straight ends. And lastly, our third example, you can see, again, is leaving sticky ends. We've got these overhanging DNA fragments. So a really common restriction enzyme that you guys will come across is EcoR1. So it's a restriction enzyme. Now, the reason why it's called EcoR1 is because it's found in E. coli bacteria. And it's a restriction enzyme that has the following recognition site. So it's going to look for GAATTC, and you'll see that it cuts between the first G and the A, and it exits between the A and the G on the bottom strand. So again, it leaves a sticky end. We can see that there'd be overhanging DNA fragments. But one thing I will point out is if you notice that the DNA running on the top strand from left to right goes GAATTC, but it actually runs the opposite way on the way back. We've got GAATTC on the back. And the reason why you might hear the terms race car and kayak, etc., is because they're palindromes, and restriction enzymes tend to cut at places that are palindromes, which means that they're the same going one way as opposed to the other way. And so often you'll see, when we look at them, that the strand of DNA will run complementary and in the opposite direction on the bottom strand. So a typical question you might get on your exam around restriction enzymes is something where you might be given a table with 4 or 5 different recognition sites. You can see here we've got 4 restriction enzymes. We've been given their 4 recognition sites, and they might give you a stretch of DNA and ask you which restriction enzyme will cut this DNA. And so what you're looking for is, where are the recognition sites? Can you find the recognition site in the DNA strand they've given you? And will it make a cut, whether it be sticky or blunt cuts? Sometimes you might get asked how many cuts are made by one recognition site or one restriction enzyme, but that's a general skill you need to know for your exam, is how to read recognition sites, how to identify where they are on a strand of DNA, and know how many cuts that they make. Now, the reason we learn about restriction enzymes is because science have taken advantage of these restriction sites. We know that bacterial restriction enzymes cut at a specific spot, and we've started to use that now to our advantage. So we now realize that because DNA is the same amongst all organisms, we can use restriction enzymes to cut all different parts of DNA, and that allows us to either insert new genes or modify them. And we can actually then glue this DNA back together using another enzyme called DNA ligase. And sometimes you'll see that referred to as DNA glue. But we've taken advantage, and we now know that we can cut DNA at spots that we want to maybe manipulate and change that DNA. So in review, restriction enzymes are produced by bacteria, and they cut DNA at specific sections. Now, those sections are known as recognition sites, and they either leave blunt or sticky ends depending on the recognition site. And lastly, we've started to use these restriction enzymes to cut DNA at desired locations, so we can then manipulate DNA after it's already been cut. So we're going to learn a little bit more about this process over the next few lessons, and how we then utilize this in technology or industry to help us achieve things in DNA that we want. So I hope this video got you off to a great start.